How's it going, folks? Can you guys hear me okay? Can you hear me? All right. Okay. All right, guys, welcome back. It's been a while. How was spring break? It sucked. It was raining the entire time. There was no spring break whatsoever, stuck inside of the home. But I hope that we're all at least safe. Second. Now, folks, we are getting close to towards the end. We're walking towards the end of the semester, and the second exam is going to be here pretty soon. So hopefully, you guys are all on top of the exam list, right? You started to study for the second exam, and then remember the third exam is going to be during the finals week. Okay, we don't have a final for this class, but we do have the third exam, which is during the finals week. And today we are covering two labs, labs 8 and lab 9. Um, and then we have only one in-lab activity, which is just related to lab 8, but it's going to count for both lab 8 and lab 9. It's going to count double, okay? Does anybody have any questions, any issues, any problems, anything at all, folks? All right. So let me share my screen with you. All right, now lab 8 is going to be talking about the special senses, and then lab 9 is going to go over the endocrine system. All right, just a reminder, the next lab is also double. We're going to go over labs 10 and 11, so which means that there are two pre-lab assignments that you need to turn in. All right, so what we're going to talk about in terms of lab 8. In terms of the lab 8, folks, we're only going to focus on eyes and ears. By the way, both of these structures will be in our next lab exam. Okay, so we're going to cover all the structures that I want you to know in regards to the eye. Then I'm going to cover all the structures that you need to know in regards to the ears. So very important lab. And then after we're done with lab 8, we're going to go straight into lab 9, which is going to be discussing about the endocrine system. Now, we know, we have seen this before in the lecture, that we can divide our eye into three different layers. We have an outer layer, we have a middle layer, and we have an inner layer. The outer layer is what I call the fibrous. The middle layer is called the vascular, and the inner layer is called the sensory. In the outer layer, folks, there are two structures that makes up the outer layer, makes up the fibrous layer. Before I show you what it is, let's do a little recap here. You tell me, can anybody tell me what is the structure in the front of the eye? This structure is translucid and allows light to go inside of the eye. What is it called? You can unmute yourself.
That's cornea.
That is the cornea. Okay, so we have the cornea. Now, what about the white of the eye? What is that called?
Sclera.
That would be the sclera. So the cornea and the sclera, they make up what we call the fibrous layer of the eye. Okay, now do not confuse the cornea with the lens, and a lot of students do that. The lens is all the way behind the iris in a chamber, in a space that we call the posterior chamber. Okay? Now, this structure, the next layer of the eye is what we call the vascular, and the vascular layer includes the choroid, it includes the ciliary body, also called the ciliary muscle, and includes the colored part of our eye, which we call the iris. Keeping in mind that both the ciliary body, aka the ciliary muscle, and the iris, they are both made of smooth muscle. We don't control them. Now, what is the function of the iris, guys? The iris is going to control the size of the pupil. What is the pupil? The pupil is the hole through the iris that allows light to go inside of the eye. Okay, and the iris again is going to control the size of the pupil, and this control is a reflex. Again, we don't control it. It is autonomic. So when the iris contracts, it's going to decrease the size of the pupil. When the iris relaxes, it's going to dilate the pupil. It's going to increase the size of the pupil. Now, we can see in the ciliary body, there are little strings that extends from the ciliary body, and then they attach themselves into the lens. These little strings are what we call the ciliary zonule. And the function of the ciliary zonule is that the ciliary zonule, together with the ciliary body, they are going to change the shape of the lens. Our lens right here, it has the consistency of a gummy bear. If you ever squeeze the gummy bear between your fingers and you can change the shape of the gummy bear, that's what the lens feels like. And the lens will change the shape depending if you're looking at something that is close to our eyes or if it's something that is far away from our eye. And it's the ciliary body together with the ciliary zonule that makes this adjustment that is called accommodation. Okay? Then we have the inner part of the eye, which is called the sensory layer, and the sensory layer is made by the retina. And within the retina, this is where we're going to find the photoreceptors that are embedded in the retina that allow us to be able to see. It is arguably the most important structure of the eye, the photoreceptors, okay? And they are found embedded in the retina. Now, there's a couple of other structures that I do want you guys to know that I will ask you on our next lab exam. One of those structures is this nerve right here, right behind the eyeball. This nerve is what we call the optic nerve. And this is going to send information from the eye all the way to the back of the brain in a region that we call the visual cortex. This is where the image is formed, the back of the brain. Okay? Another structure that I want you guys to know is this little region right here inside of the eye. I just circled for you in red. This is what is called the optic disc, aka the blind spot. What is this optic disc? The optic disc is a region inside of the retina where the light is not perceived because there are no photoreceptors on the optic disc. So what is the function then of the optic disc? The function of the optic disc is that all the axons of the photoreceptors in the retina, they're going to leave the retina, leave the eyeball, and they are going to bundle together to form the optic nerve. So you can think of the optic disc, the exit point of the photoreceptors inside of the eye. And we call it the blind spot because when the light hits the blind spot or the optic disc, we cannot see that light. Why not? Because there are no photoreceptors. Okay? And finally, all the way embedded in the retina, we have this little region right here that we call the fovea centralis. What is so special about the fovea centralis? The fovea centralis is where we find the highest concentration of photoreceptors in the retina. So when we stare straight at an object, the light is going to go straight into the fovea centralis. And this is where we see the sharpest. Okay? Now, it doesn't mean that we don't have light hitting outside of the fovea centralis. But outside of the fovea centralis, we are not going to see as sharp because we have less photoreceptors. Now, we can actually do a test for the blind spot, and all that you need is an index card. And in this index card, kind of like this one here, and in this index card, you need a dot on one end, and a couple of inches away, you're going to have a cross there, a little plus sign. And then you're going to test one eye at a time. You're going to close one eye and stare at the plus sign, and then you're going to move the card closer to your eye. There's going to be a distance from your eye in which the dot right here disappears from your peripheral vision. Because at that distance, the light that is hitting the dot and coming inside of your eye is hitting the optic disc. And if it hits the optic disc, we can't see the light. Okay. Now, we do have two different types of photoreceptors in the retina. We have the rod cells and we have the cone cells. There is only one type of rod cell, folks, but there are three different types of cone cells. We have the green cones, the blue cones, and the red cones. Now, rod cells are involved with our dark vision, and we're going to explain to you why in a minute. While the cone cells, they're involved with our color vision, okay? Now, why are those different cones, they have different names? Well, because they are excited by different wavelengths of light. That is it. The blue cones are excited by wavelength of light within the blue range. The green cones are excited by wavelength of light within the green range, and the red cones are excited by wavelengths of light within the red range. Okay? Now, some people, they are colorblind, and that is because they might have a decrease in the number of cone cells. It could be a decrease in the blue cones, a decrease in the green or the red, and they're going to have a harder time seeing those colors. What about the rod cell? The rod cells, guys, they are involved with dark vision because they are the most sensitive photoreceptors to light. And the example that I always give is when you wake up in the middle of the night and you open your eyes, you will not see color in your room. You're only going to see shades of gray because the amount of light in your room, dark, in the dark at night, is not going to be enough to excite the cone cells, but it is enough to excite the rod cells. Okay? Now, some people have a night blindness. What is night blindness? Night blindness is when you can't see very well at night. And that is usually associated with a lack of vitamin A, because vitamin A is essential for the formation of what we call the rhodopsin. And the rhodopsin are the proteins that become excited within the rod cells. So if you don't have enough vitamin A, you cannot make enough rhodopsin, your rod cells will not become activated. Okay? Guys, here we have a simulated vision of a human being and the simulated vision of a cat. Now, cats, dogs, cattle, they're all active at night. And therefore, they have a better night vision than us. We can see right here on the top, which is simulating a human vision, we can pretty much just see whatever is right under the light, right here. We cannot see anything outside of the light. But if you look at a cat's vision at night, we can see that they have a much broader field of vision at night. They can see a lot better. Now, why that is the case? Well, the reason why animals that are active at night, they can see better than us, is because inside of the eye, they have a structure called the tapetum lucidum. So right here, we're looking inside a cow eye. Okay, this is a cow eye dissection. And we can see this bluish, greenish, iridescent membrane. This is the tapetum lucidum. Now, what happens is that when the light enters the cow eye or a cat's eye, it's going to bounce around this reflective surface here, and it's going to go back into the retina. So it's going to pass more than once through the retina. And because of that, they're going to be able to see better at night. Human beings don't have a tapetum lucidum. Okay? Now, let me show you some of the structures of the cow eye, and this is one of the things we're going to do today. We're going to watch a little video on the cow eye dissection. And we share a lot of the same structures with cattle and other animal. You know, we can see here in the sagittal section, we can see the lens. We can see this cream color structure right here, which is the retina. Underneath, we can see a little bit of the tapetum lucidum in a bluish color here. We can see the cornea in the front. We can see the iris that forms the pupil right there. So we share a lot of those structures, okay? Now let's watch a little bit of this dissection here of the cow eye. And this is going to be a very important dissection because again we share a lot of the structures with humans as well. Okay. That's also what we call the ciliary zones. Okay. Any questions, guys? All right. So let's move on then to the next structure that will be on our lab exam, and that is the ears. And again, we're going to walk you through all the structures that you need to know for the exam. By the way, folks, I get this question often. I didn't get it this time, but I get it often. Remember that both the eyes and the ears are bilateral, and you know what that means. Okay, means that you need to tell me if I'm looking at the right or if it is the left. Now, what if I have an eyeball, just like the eyeball that we saw, the dissecting eyeball of the cow, that is just sitting on a table? Can you tell if it is right or left? No. But if it is an eyeball that has on the skull, there are some features of the skull, as long as you can determine right from left, then you need to say if you're looking at the right or if you're looking at the left. Okay? You're going to be the right or left lens. Is the right or left sclera? Is the right or left iris? So you need to put that down in the answer. The same apply for the ears. You will be able to determine if you're looking at the right or the left. Can anybody tell me on the screen right here, right now, are we looking at the right or the left ear? See here? Is the right. Absolutely. There is the right, okay? Now, let's take a look at some of the structures. We can divide the ears into three main areas or three main sections. We have what we call the external ear, the middle ear, and the inner ear. The external ear, we're going to find what we call our ears, which is this flap of skin and fat and cartilage. This is called the auricle or the pinna. Okay? So this would be, if I was pointing to this one here on the exam, this would be the right auricle or the right pinna. Now, what is the function of this auricle? Do we have it so that we can hang our glasses or put earrings on? No. The function of the auricle or the pinna is that it's going to serve as a funnel to funnel in what? Sound waves. The sounds waves that are coming from the environment, they're going to hit the auricle, and then they're going to be guided inside this canal here called the external acoustic meatus, or simply called the auditory canal. All right. Now, this auditory canal, at the end of the auditory canal, we're going to have this membrane right here. This membrane is called the tympanic membrane. And when the sound wave hits the tympanic membrane is going to vibrate. Now, what does that vibration do? The vibration is going to amplify the sound waves. And in the middle ear, we're going to find the three little bones, the smallest bones in our body. These are what we call the auditory ossicles, but each one of them, they have a name. So we have the malleus, we have the incus, and we have the stapes. You can see here that the malleus is directly attached to the tympanic membrane in the middle ear. So when the tympanic membrane vibrates, guys, the malleus is going to vibrate as well. And then the vibration is going to be sent to the incus. And then from the incus, the vibration is going to go to the stapes. Okay? So now the stapes is attached to another bone in the inner ear, and this bone is called the vestibule. And the vestibule, together with what we call the cochlea, they are filled with fluid on the inside. So when the stapes right here, when the stapes vibrate, it's going to cause the vibration of the fluid inside the vestibule and inside the cochlea. And the vibration of the fluid will cause little waves to start to form, guys. And those little waves, they're going to travel from the vestibule into the cochlea. And inside of the cochlea, we have the spiral organ of Corti. We're going to take a closer look at it in a minute. And this is where we perceive sound. Now, we can see that right behind the cochlea, there is a nerve right here. This nerve is going to send sound information to the brain. And this nerve is what we call the cochlear nerve. Okay? Now we can see that there's another nerve right here. And this nerve is attached to the vestibule and attached to the semicircular canals, which are these channels right here on top. This nerve that is attached to the vestibule and the semicircular canals is what we call the vestibular nerve. And the vestibular nerve is involved with our sense of equilibrium. Because our ears is not just for hearing. Our ears are also involved with our equilibrium, with our balance. Okay, and we're going to see there are two different types of balance. We have static equilibrium and we have dynamic equilibrium. We're going to talk more about it in a minute. And finally, the last structure you guys need to know is this tube right here that connects our middle ear with our throat. Yes, there is a direct connection between the throat and the middle ear. And this is what we call the pharyngotympanic tube, also called auditory tube or Eustachian tube. Do not confuse auditory tube with auditory canal. If you mix the two, you are naming the wrong structure. All right. Any questions, folks? All right. Moving on. So let's take a closer look at the cochlea now. And as I mentioned earlier, inside of the cochlea, this is where we're going to perceive sound. So if I open up a cochlea, this is what it would look like. We see all these chambers right here. These chambers are filled with fluid, and inside some of these chambers, we're going to find a little apparatus called the spiral organ of Corti. And the spiral organ of Corti is where we perceive sound. So we can see how we have our cochlear nerve directly attached to the spiral organ of Corti. Now, within the spiral organ of Corti, we have this membrane called the tectorial membrane. And underneath the tectorial membrane, we have little cells, little sensory organs called the hair cells. They have nothing to do with hair. They're just called the hair cells. What do? When the little wave that is propagating, that is going through the cochlea, when it gets to the spiral organ of Corti, the tectorial membrane will also undulate. As they undulate, the little hair cells underneath is going to become activated. They are going to generate action potential, and the action potential is going to be spread through the cochlear nerve, sending sound information to the brain. Okay? Now, there are two different types of deafness. Since we're talking about hearing, we have what we call conduction deafness, and we have something called the sensory neural deafness. Conduction deafness is when we have a problem with the conduction of the sound wave. It could be a big earwax inside of your auditory canal. It could be that your tympanic membrane is damaged. It could be that your auditory ossicles are broken, and that is going to stop the propagation of the sound. Another type of deafness is called the sensory neural deafness, and that one is more serious and more permanent. This one has to do with the cochlear nerve and with the little hair cells. Now, the last part on this lab right here, I want to talk about equilibrium, okay? Which is another responsibility of the ears. And as I mentioned earlier, we have both static and dynamic. Static equilibrium is when we're standing still. Dynamic equilibrium is when we are spinning around, walking, running, moving our head. The static equilibrium is perceived inside of the vestibule, right here. If I open up the vestibule and I look inside it, it is hollowed, filled with a fluid. On top of the fluid, there are little calcium stones called otolith, and embedded in the fluid, I have little sensory cells called the hair cells. When we stand with our head upright, guys, all the otolith are horizontal. The little hair cells are standing upright. When I tilt my head forward, now the little hair cells are also going to tilt forward because gravity is going to pull on the hair cells. The bending of the hair cells forward will cause them to become activated, and they are going to send information to our brain, telling the brain the position of our head. Okay? Now, what about the dynamic equilibrium? Dynamic equilibrium is perceived in a different spot. It is not perceived in the vestibule right here. It is perceived at the joining of the vestibule with the semicircular canals. This region here is called the ampulla. This is where dynamic equilibrium is perceived. And in dynamic equilibrium, as we spin around, folks, there is a membrane called the cupula that is very similar to the tectorial membrane. And this cupula here, as we spin, the cupula will also spin in the same direction. As the cupula spin, the little hair cells that are embedded in the cupula, and we can see the little hair cells here, they will spin in the same direction at the same speed. And they are going to become activated, sending information, dynamic equilibrium information to the brain. Okay? So that we don't fall over when we are dancing or ice skating, whatever is that you do, right? Any questions, guys? All right, folks. That is it for the eyes and the ears. Let's go over the endocrine system. And the good news is that the endocrine system is not going to be in the lab exam. Okay, but still, I do want to cover, but there is no question about the endocrine system in the lab exam. Now, let's start quickly here, making a comparison between the nervous and the endocrine system. We have talked about both of these, in fact, before in the lecture. We know that the messenger for the nervous system are the neurotransmitters. They are going to target the skeletal muscles, the smooth muscle, and glands. And the neurotransmitters have a short-term duration. They don't last very long. The endocrine system messengers are the hormones. They target pretty much our entire body, and they can last a long time in our bloodstream. They have long-term duration. Okay? We also know that we have two different types of hormones. We have the lipid-soluble and we have the water-soluble. We know that the lipid-soluble is going to cross the plasma membrane because it's made of phospholipid. It's going to go all the way inside of the cell and it's going to change the transcription level of a gene. It's going to cause transcription that then is going to cause translation and the formation of a protein. Water-soluble hormones, they do not cross the phospholipid bilayer. And because of that, they need a cell surface receptor, which will activate enzymes that is going to lead to the formation of a secondary messenger called the cyclic AMP. And the cyclic AMP will lead to the phosphorylation of proteins. So water-soluble hormones act at what we call the post-translational level, while the lipid-soluble, they act at the transcriptional level. So let's go over some of those major endocrine organs, starting with the pituitary gland. Now, the pituitary gland is divided into two lobes, and I need everybody's attention here so you don't make this mistake in the quiz today. The pituitary gland is divided into what we call the anterior lobe, which is the bigger one right here, and the posterior lobe. There is no right and left pituitary gland. There is anterior and posterior. Okay? And the pituitary gland, they are controlled by another gland in the brain called the hypothalamus. So the hypothalamus controls the function of the anterior pituitary and the posterior pituitary. Now, the anterior pituitary produces several hormones that will control the activity of other glands and structures in our body. And for that reason, we call the anterior pituitary the master endocrine gland. Okay? Some of the hormones that the anterior pituitary produce includes the human growth hormone that will control the growth of our bones and muscles, the prolactin that will control the production of milk by the mammary glands, the luteinizing hormone and the follicle-stimulating hormone, which will act on our gonads, the thyrotropic hormone that will activate the thyroid gland, and the adrenocorticotropic hormone, which will activate the adrenal cortex. Okay? These are all hormones produced by the anterior pituitary. Now, the pineal gland, which is a little gland all the way in the epithalamus of the diencephalon, right here, it is responsible for the production of a hormone called melatonin. And some of us might be familiar with melatonin. In fact, you might even take over-the-counter melatonin. And melatonin is involved with our sleep and wake cycle. It helps us decrease our metabolic rate at night so that we can fall asleep. Okay? The thyroid gland produces two different types of hormones. It produces what we call the thyroid hormones, and it produces another hormone called calcitonin. The thyroid hormones are produced by these little cells here called follicular cells. So they produce what we call the T3 and the T4 hormones, which collectively are called the thyroid hormones. The parafollicular cells, they produce a different hormone called calcitonin, okay, which is responsible for bringing the level of calcium down in our blood. Behind the thyroid gland, we're going to find another gland called the parathyroid gland. Can you guys see the little green nodules right here behind the thyroid? These are what we call the parathyroid glands. And the parathyroid gland, they produce a hormone called the parathyroid hormone, or PTH. And PTH just raises the level of calcium in the blood. Okay? It is what we call a hypercalcemic hormone, while the calcitonin is a hypocalcemic hormone. The adrenal gland is a very important gland divided into two sections. There is the outer layer of the adrenal gland called the adrenal cortex, and the inner section of the adrenal gland called the adrenal medulla. The adrenal cortex produces mineralocorticoids and glucocorticoids and a little bit of androgens and estrogens, while the adrenal medulla, they produce collectively what we call the catecholamines, which include epinephrine and norepinephrine. And these hormones right here, guys, these are our fight or flight hormones. Okay? They are released when we are under stress. The pancreas is not only an organ of the digestive system, but is also an endocrine organ as well. And the pancreas produces two different types of hormones. It produces what we call the insulin and the glucagon. Insulin is produced by the little beta cells of the pancreas. Glucagon is produced and released by the alpha cells. Glucagon increases blood glucose level. Insulin decreases blood glucose level. Okay? And finally, the ovaries and the testes, which are what we call our gonads, they are also major endocrine organs. What kind of hormones they produce, guys? They produce the sex hormones, which are estrogen, progesterone, and testosterone. All three of these hormones are based on cholesterol. If you don't have enough cholesterol in your body, you will not be able to make the sex hormones, okay? Because the backbone of estrogen, testosterone, and progesterone is cholesterol right here. Okay. All right, guys, so what are we doing today? Today we have two videos on Edpuzzle. One video is going to walk you through the model of the human ear. The second video is going to walk you through the model of the human eye. Remember, I will use those models, pictures of those models for the exam, so you need to familiarize yourself with them. After you're done with those videos on Edpuzzle, then you're going to jump back on Lamaku and you're going to take the quizzes. And there are two quizzes open for Lab 8 and Lab 9. Okay? Let's get started.